Hi everybody, my name is Andrei Deviatkin. I'm working at Sechenov University and I'm glad to present you results of my work. And we are talking now about multiple long-distance transfers in TBEV spread pattern. TBEV is a tick-borne encephalitis virus, and approximately 20 years ago, it was subdivided into three phylogenetically distant subtypes of this virus, and they were called European TBEV subtype, Siberian TBEV subtype, and Far Eastern TBEV subtype. And at this map, it is shown that in the end of 90s, there were just approximately 60 sequences known at this moment, and mostly Siberian TBEV subtype were found in Siberia. Far Eastern TBEV viruses were located in the Far East, and European viruses were found mostly in Europe. But, 20 years ago, the phylogenetic tree of all known viruses was rather small, as it was demonstrated here. Yes, but now, due to the emergence of new high-throughput sequencing technologies, there are thousands of these sequences, and the picture seems to be much more complex than it was supposed 20 years ago. There are a lot of, at first, the area of distribution of tick-borne encephalitis virus currently differs from that it was several decades years ago, and now it was recently indicated that the TBEV was found in the United Kingdom, in Denmark, in France. Previously, it was not shown. Also, in Moldova, for example, we can find Far Eastern subtype of tick-borne encephalitis virus, and here it is not shown, but for example, in Crimea Peninsula, we can find Siberian, Far Eastern, and European subtype. Recently, Siberian subtype of TBEV was found even in Bosnia and Kazakhstan. And currently, it seems that the real distribution of this virus in different geographic parts of the world is completely different from as it was supposed 20 years ago. And in my work, I have downloaded all sequences of TBEV virus. And here we can see that the genome regions of this virus are sequenced very uneven. And approximately totally there are a little bit more than 2,000 sequences. And the region of here gene, it is a gene which encodes an envelope protein, is sequenced most often in comparison to all other parts of the TBEV genome. For example, there are approximately 200 of viruses with complete or near complete genome sequenced, and approximately 800 viruses that were sequenced at this E gene protein. And for my analysis, I have used these E gene sequences. At first, we have tried to simulate the situation as it was known approximately 10 or 20 years ago, when there were not so many sequences. And here we estimated substitution rate, and for different sequences, different data sets of sequences. They were chosen randomly, and there were 10 data sets with 50 sequences and 10 data sets with 100 sequences, randomly chosen from these 800 sequences. And we can see that when we use a data set consisting from just 50 sequences, the substitution rates are very different. And this means that if this work was done in the period of time where there were not known a lot of sequences, the results were very different, and they are not even interacting with each other. And even if we use 100 sequences in our analysis, the results can be also not so close to each other, but at least they are overlapping. In the left panel of this slide, it is demonstrated this inconsistency in substitution rates, and in the Right panel of the slide, it is shown the median root height in years. It means when the most recent common ancestor of these 50 or 100 viruses existed according to some Bayesian estimates. And the most actual tree that was made by me approximately a half year ago, it was consisted from 551 viruses. And it is the most represented genome fragment of TBEV viruses. And we can see, yes, there are three main subtypes. They are currently called European, Far Eastern, and Siberian, but they are not located only in Europe, Far East, or Siberian. And also, two or maybe three novel putative subtypes were proposed recently. Two viruses were found in Chinese Tibet, in Himalayan. It is recognized as a novel subtype. And two other group of viruses were found near Baikal, and they are supposed by some researchers to be novel subtypes too. But it is not clarified at this moment. Maybe they will be regarded as a novel subtypes. And when we have tried to analyze different subtypes separately, we can see that different geographic regions are phylogenetically connected. At this phylogenetic tree, sequences were colored according to geographic region of isolation, and all the world, all the TBEV distribution areas were subdivided by me for different geographic regions. For example, at this map, by the red color, viruses from Germany, Switzerland, Austria, Hungary, Czech Republic, Slovakia, and Slovenia are colored by the red color. And for example, sequences from Altai region are colored from this sandy color. And we can see that viruses that phylogenetically very close, for example, Altai and St. Petersburg viruses that were isolated from Altai region of Russia and St. Petersburg region of Russia. There are thousands of kilometers between these viruses, but it's strange that they're located together because suppose that tick-borne encephalitis is distributed by its vector, and it may be it's supposed to be a tick. And tick in Altai and in St. Petersburg are different ticks. The same situation we may see in Siberian subtype, because Siberian subtypes currently can be found in very different regions. For example, it was found in Tuva, in Kazakhstan, in Yekaterinburg, in Kyrgyzstan, in Kemerovo, in Vologda, Karelia, in Estonia, in Baltic countries. And the picture here is the same as for European TBEV subtype. And the same situation is for Far Eastern subtype of TBEV virus, because some viruses are isolated from Ukraine, and they are phylogenetically related to viruses isolated from Vladivostok, and it's very strange. Estonia and Vladivostok, there are a lot of such strange locations. And another point that I want to indicate is that pairwise genetic distances for most part of European subtype TBEV viruses, there are approximately 200 viruses, is 3% of identity. That means that every two pairs of viruses differs from each other. In mean, it is 2.7, 2.8% by its identity. And the same situation is the same for all European subtypes that is distributed in Irkutsk Oblast, in Irkutsk region, in Germany, in Austria, in South Korea, for example. And the distribution of these pairwise genetic distances is the same that for viruses isolated only in Switzerland, for example. There were, in my data set, there were approximately 41 TBEV viruses that were isolated in Switzerland, and the distribution of these pairwise distances was the same as for whole European subtype. And the same situation was for Czech Republic, for example. And it seems to be a pattern for spreading of this virus and not something strange and anecdotal. And also, we have chosen some pairs of viruses that were phylogenetically very related and estimated their time of existence of the most recent common ancestor of these two viruses. And for example, there were some viruses, for example, this one, MH6634280, 428 was isolated in Moscow, in Krylatskaya, in 2017. And the most closest, according to Bayesian phylogenetic tree virus, was isolated in Slovenia approximately 20 years ago. And so there are approximately 2% of identity difference. And yes, but it means for this virus that most recent common ancestor between these viruses existed very many years ago, but approximate direct distance between Moscow and Slovenia is approximately 2,000 kilometers. And for example, some other viruses are much more closer in terms of phylogenetics. For example, virus from Latvia and Germany, it is approximately 1 and 2 substitution in this 1,000 nucleotide region. And they were isolated in the territories that are situated in, there is approximately 1,000 kilometers between these two countries. And the most common ancestor for these viruses existed just 50 years ago. And the same situation is for Siberian TBEV virus. Some viruses are identical. For example, virus isolated in Sakhalin in 2011 and virus isolated in Chita in 2000. They are completely identical. But the geographical distance between Sakhalin and Chita is approximately 2,200 kilometers. And there is just 20 years of difference. Yeah, it's a little bit strange, but it is the result of this Bayesian estimates for common ancestor according to all other sequences in this data set. Yes. And the same situation was indicated for Far Eastern subtype of TBEV virus, because there are some identical viruses isolated in Khabarovsk and Tomsk, for example. And it seems to be that long transfers may be considered as normal and an abundant pattern in TBEV spreading. Thank you very much for your attention. This research was funded by Russian Science Foundation. Bye-bye. Have a nice day.